All right, thank you everyone for joining us today. Just had a few brief announcements. We hope everybody will be able to come to the Early Detection of Cancer Conference this year, which will be virtual and be held in October from the 6th to the 8th. And you can see the registration for your interest link is also here. So we'll look forward to seeing you there. And also just for our upcoming seminar we'll have from Dr. Raza, and that will be on October 12th. This should be another really interesting seminar. And so for our speaker today, I'll hand it over to Utkan. Please go ahead and introduce it and take it away.
Thank you, Ryan. I would like to thank Dr. Michael Berger for joining us today. He is the Elizabeth Felix Rohatyn Chair and Associate Director of the Marie-Josée and Henry Kravis Center for Molecular Oncology at the Memorial Sloan Kettering Center, Cancer Center. He's also an associate attending geneticist in the Department of Pathology. His expertise spans cancer genomics, computational biology, high-throughput DNA sequencing technology, and his lab's work develops experimental and computational methods to characterize the genetic makeup of individual cancers, and thereafter identifying genomic biomarkers for drug resistance and drug response. And they've done really interesting studies, and he will be holding and sharing his work and some of the work they have done recently, as he outlined in his abstract. And one thing to note is Dr. Berger holds a bachelor's degree in physics from Princeton and a PhD in biophysics from Harvard. We welcome you, Dr. Berger, and thank you for joining us.
Right. Thank you, Utkan. I hope I can live up to your generous introduction. And it's, hopefully just let me know if this isn't displaying properly, but I hope it is. Still weird to be, giving talks from, my office by myself. I wish I could be with you in person. And hopefully there'll be opportunities for some questions at the end and some informal chatting later in the day. But I'm going to talk to you today about some of the work that we're doing here. Title of the talk, How Tumor, Normal, and Plasma DNA Sequencing Are Informing Cancer Care, Cancer Risk, and Cancer Detection. I'll be honest, I should admit, I probably have the least to say about cancer detection, and I know that's the sort of topic that brings this group together. But it is an area of significant interest, and I think through our clinical genomics program, identifying populations at greater risk for cancer is providing us an opportunity to explore some of the technologies that we're developing and their utility for cancer detection. We are a cancer center here at Memorial Sloan Kettering, so most of the samples that we study are from patients who've already been diagnosed, but there are efforts to try to bring the treatment and intervention earlier and earlier, which depends on being able to diagnose cancer at its earliest stages. Here are my disclosures, none of which are directly relevant to any of the data or analyses that I'm presenting. So I will be talking about two different next generation sequencing-based clinical tests that my lab's developed that we're running in high throughput here at Sloan Kettering. One that's been around for many years longer, called MSK-IMPACT, allows us to profile genomic alterations in tumor tissue. And more recently, we've developed and implemented a plasma-based cell-free DNA test called MSK-ACCESS. I'll describe both of these as they come up and start with MSK-IMPACT. So this is what's, I guess, now become fairly standard processes where we isolate DNA both from a patient's tumor tissue, could be a resection from surgery or biopsy, as well as blood, which in this case, the white blood cell DNA is used as a normal control. So initially to subtract out inherited variants and provide information about the truly acquired somatic mutations in the cancer, although we're learning more and more from analyzing the white blood cell DNA itself. But this is not plasma cell-free DNA sequencing. This is just a source of normal. Paired DNA libraries from both samples sequence to deep coverage, typically around 700 to 800x coverage in the tumor, and about 400 to 500x coverage in the matched normal, and called several different classes of alterations. So this is a test that's currently comprised of 505 genes, where we're looking for mutations, indels, copy number alterations, gene fusions, and now more complex signatures like microsatellite instability, tumor mutation burden, so on. And have a team of over 20 bioinformaticians in our department of pathology working with molecular pathologists and clinical fellows to interpret and report these alterations. All of this is stored in a database that feeds our clinical reporting as well as clinical trial matching and research initiatives. I will talk about a little, some of this in a little more detail, but just to give you an overall historical perspective on our test, something that we established in my lab first now 10 years ago for translational research projects to retrospectively study tumors collected from clinical trials or rare cancer types and better understand the genomic drivers and their relationships to outcomes into therapy. Began validating this for clinical applications in our molecular diagnostic service and pathology a couple of years later. And since 2014, it's been approved by the New York State Department of Health for clinical purposes. So this has really sort of transformed the clinical care that's provided and integrated this genomic profiling as part of the clinical decision-making for the last 7 years. Shortly thereafter, we created, through a research protocol, an opt-in consenting mechanism for clinical germline analysis, which I'll describe, which is how we define these higher risk individuals. But sometimes these germline alterations are directly relevant to treatments that patients may benefit from as well. And we were the first test to obtain FDA approval as a class 2 tumor profiling assay, academic or commercial. And last year we passed 50,000 patients profiled. Now it's about 63,000 patients using this panel. Just to give you a very brief sort of high level overview for this clinical program, when we detect mutations, and we can detect a handful to dozens to sometimes hundreds of mutations in a single tumor, we need this to be sort of interpretable, clinically digestible by oncologists and surgeons increasingly who are ordering this test on their patients. So we rely on a knowledge base called OnceKB, which was a large initiative led here by Nikki Schultz on the informatics side and Devyani Chakrabarty in collaboration with many, many clinicians and clinical and research fellows to sort of curate and aggregate the available information about the clinical and biological significance of genomic alterations, not just at the gene level, but at the specific alteration level and within individual cancer types, using some of the resources here, but really the full breadth of literature and scientific knowledge. There's a whole tiering system. This is similar to other commercial and academic knowledge bases in their efforts to aggregate this information, because this is a challenge that the whole field faces. And from our analyses, just over half of patients have at least what we call a level 3, so compelling clinical evidence, but many a level 2 or level 1 actionable mutation in their tumor. And this has grown. The first study that we did on the first 10,000 patients who had received MSK-IMPACT profiling in 2017 showed this closer to 37 to 40%. Now it's over 50%. And this incorporates new targets, new drugs, but also clinically relevant signatures like microsatellite instability, which account for an additional fraction of patients where the sort of clinical significance is not for targeted therapy, but for immunotherapy. And these get reported back to patients through our clinical reports as well. So all of this data can be integrated then with other diagnostic data, clinical data, demographic data, financial data, scheduling data, the whole data warehouse, it's called the Darwin Institutional Database, to facilitate automated alerts to clinicians about patients with alterations that might qualify them for clinical trials. And this is particularly important now with a greater number of what we call basket clinical trials. Some of you may be familiar. The idea being that patients can be enrolled into a single basket trial across all types of cancer, whereas in the past they might have been limited to patients with only lung cancer or only ovarian cancer. But what's unique about basket trials is that patients have to harbor a specific alteration or in a gene or pathway that is targetable by the drug that's being studied. So all patients with all types of cancer might be eligible based on the presence of a shared alteration or mutations in a given gene. And this has allowed us this sort of automated and integrated informatics system to screen patients for the subset that might be the best candidates for particular targeting therapy clinical trials. And here are several examples of multi-institution basket trials for a HER kinase inhibitor, neratinib, a TRAC inhibitor, larotrectinib, RET inhibitor, selpercatinib. Some of many examples where the trials were open at 10 to 20 different centers, but because of this large-scale profiling effort that we had implemented, decoupled from any individual therapeutic trial, but as a standalone prospective clinical genomic profiling effort, we can build a database of patients and alterations that feed into all of the precision oncology trials that were available. And in each of these cases, enrolled the majority of patients for the whole study, but just being one of many different sites. And this allows the oncologists at Sloan Kettering to really drive these studies and provide the academic leadership of these investigations. Altogether, as I said, we've sequenced over 63,000 tumors from over 54,000 patients, and have really emphasized data sharing as part of our program. So all MSK investigators have real-time access to the full de-identified set of data from MSK-IMPACT. And this has led to, at this point, actually over 400 papers published by MSK investigators featuring the clinical MSK-IMPACT results. And we use the CBio portal developed by Nikki Schultz's team as a way of disseminating those results internally. These papers have spanned the full spectrum of research and clinical departments and really been a boon for researchers who might not have even been interested in genomics, and even basic research labs that are developing mouse models and doing in vitro experiments, but providing that data that can inform a particular model or prioritize mutations for functional characterization based on what we actually see in patients. But we've also prioritized data sharing globally through the AACR Genie initiative. So this is the latest release through AACR Genie. This is a program run through the American Association for Cancer Research of centers that are doing local clinical sequencing, the MSK-IMPACT panel being the most comprehensive of the panels that are being used, and actually one of the only ones that uses tumor and matched normal DNA. So one of the challenges with AACR Genie is that there's a whole diversity of panels and platforms and analysis pipelines that are used. But there's this core set of genes that are shared across all of them. But the MSK-IMPACT data comprises over 40% of the entire data set, which actually seems to be growing and not shrinking as we move the program forward. But what I wanted to focus on, for the next section is the additional genomic features that we can detect. So everything I've emphasized, aside from the microsatellite instability signature, has relied on the detection of a single mutation and really just the presence or absence of that single somatic mutation. But the fact that we're generating so much data from both tumor and normal DNA from each patient allows us to really extract a lot more information from the data that we generate. And just say a little bit more about the somatic mutations. The list on the left are what we actually report back in our clinical reports. And again, it's really just the presence or absence of a given alteration type or a given signature. But what we've begun to do through research initiatives in my lab and other collaborating labs in the Center for Molecular Oncology is try to expand the breadth of alteration types and properties of mutations beyond just presence and absence that may have additional clinical or biological significance. So this could be a whole talk in itself, and it really is a whole talk in itself that maybe I can deliver another day. But these lists of allele-specific copy number, loss of heterozygosity, whole genome doubling, these appear to have prognostic significance as we can detect these features in our now data set of over 60,000 tumors, correlated with the outcomes and drug response data that exists for these patients. In addition to the presence or absence of mutations, we can look at the zygosity and allelic imbalance or the clonality of mutations in combination with clinical data to understand where homozygous mutations or amplified alleles may behave differently than balanced or heterozygous mutations, or clonal mutations versus subclonal mutations. We can infer HLA genotypes and antigens from the data, and we're beginning to perform studies around genetic ancestry inferred from the MSK-IMPACT data. I want to say a little bit more about germline variants. So this is where we can identify cancer risk, and this has implications for some of our efforts in cancer detection as well, because these patients are at significantly greater lifetime risk of cancer. Either they're patients who have cancer where we detect a germline alteration that might be at greater risk of developing a second primary tumor, or they're relatives of patients who then through follow-up screening and testing might now know that they're carrying the same germline variant that puts them at greater risk. So we have a whole program now. I mentioned in 2015, we started performing opt-in germline testing. And the way this works is within our Department of Pathology, there's a team of molecular pathologists specialized in genetic alterations and PhD level scientists who can provide the interpretation and clinical curation. Members of our clinical bioinformatics team that oversee the pipeline, working in collaboration with the medical geneticists and genetic counselors in our clinical genetics service. In the past, genetic testing always required a full visit with a genetic counselor for pre-test counseling to discuss the risks and benefits of what might be learned through this testing. This has now moved into a short 6-minute video that patients watch in their oncology clinics, such that we can perform population scale screening, and then when patients, a subset of patients have a positive germline result with pathogenic germline allele, that would then trigger follow-up with our clinical genetics service. Our experience now is about 20,000 cases of patients who opted in for this what we call secondary germline analysis, where there may not be any known family history or reason for suspecting a pathogenic germline allele. They're just having their tumor sequenced with the matched normal alongside of it. But in this population, almost 20%, 18% have been discovered to have either a pathogenic or likely pathogenic germline variant that's then triggered a follow-up appointment with clinical genetics. And you can see on the left in blue the distribution of cancer types within this cohort. And on the right, the rate of detection of pathogenic or likely pathogenic germline alteration. So I'm going to say a little bit more about this in a few slides. But I want to also introduce clonal hematopoiesis. So these are, as many of you may know, somatic mutations in hematopoietic cells. So they're present in the blood, but not the tumor, in the same genes that are associated with leukemia, but not necessarily in patients with full-blown leukemia. But they lead to a clonal expansion of the hematopoietic cells that harbor the mutations. And these can be precursors to hematologic malignancies. So it's sort of another form of cancer risk that we can infer from the tumor normal sequencing. And just to say a little bit more about this, we published a study a few years ago now looking at the first 8,000 patients where we had MSK-IMPACT. This is an analysis led by Amit Zahir and clinical bioinformatics and Callie Poons from medical oncology, where we identified mutations associated with clonal hematopoiesis in as many as 30% of patients in our solid tumor cohort. This was higher than what had been previously reported through analysis of TCGA data and 1,000 genomes data, in part because this is a slightly older population, and we know that clonal hematopoiesis is strongly associated with age, but in small part because of the effect of prior cancer-related therapy, as I'll show you. But I think largely because this is deep coverage sequencing. I mentioned that we sequence and match normal about 400 to 500x coverage, which gives us greater sensitivity for detecting clonal hematopoiesis at the 1% to 2% levels that we more often observe than 20 to 30%. And these genes that are mutated typically are associated with where mutations are associated with leukemia and other hematologic malignancies. This is just to compare the age distribution of germline variants versus clonal hematopoiesis mutations. The germline variants in gold. You can see that the gray bars represent the number of patients within each age bin within the cohort. And the lines are on the right axis, the proportion within each age bin that harbor the germline variant or clonal hematopoiesis. And for germline, you can see, if anything, is a trend downward, where the germline carriers are typically a little bit younger on average, whereas clonal hematopoiesis, there's a dramatic increase in frequency as you get to elderly age distributions. And when we look at associations now, since we have this population scale data with prior cancer therapy or tobacco use or or other, within therapy, there's chemotherapy and radiation therapy where we see this association. There are these positive associations with clonal hematopoiesis. Patients with clonal hematopoiesis have shorter survival on average, even for their solid tumors, suggesting there may be an effect on how patients may respond to therapy, or the effect that therapy itself may have on certain subclones within blood. And most significantly on the right is a very increased risk of a subsequent hematological malignancy for patients with clonal hematopoiesis versus not. And that final finding, because of the increased risk, led to the establishment of a new first of its kind clinic within MSK of patients who have incidental findings of clonal hematopoiesis from MSK-IMPACT tumor normal sequencing. And if we detect clonal hematopoiesis and they have abnormal blood counts and they have an otherwise favorable prognosis of their solid tumor, so not necessarily late stage pancreatic cancer, but patients that might have earlier stage disease with a good outcome predicted, those patients would be referred for sort of serial monitoring given their increased risk of developing leukemias. And this clinic, led by Ross Levine, has actually led to early detection of malignancies in a number of patients that are then immediately referred to the leukemia service. So I think it's sort of a little bit of a tangent, but I think relevant to the topics that I'm discussing is a major area of interest in our lab, which is how these germline variants influence somatic phenotypes, or to what extent do germline variants that we detect contribute to the somatic phenotypes either in the tumor or in clonal hematopoiesis and the progression of cancer, and how often are the tumors that we detect in carriers of germline alterations dependent on those germline alleles and pathways. So I'm going to spend a few minutes talking about work that was really spearheaded by Preeti Srinivasan, who's now with you. She's a scientist in Christina Curtis's lab. We're sorry to see her go, but she had a very successful PhD as a graduate student in my lab, and worked alongside Chai Bamamudi in Barry Taylor's lab when Barry was here at MSK. So I'm going to present their work briefly, all unpublished still, and then a little bit of Sebas Franch Exposito's work. He's a postdoc in my lab working on clonal hematopoiesis. So what Preeti and Chai had done was take all of the data, not just the patients who opted in and consented, but create this cohort-wide anonymized resource of germline variants across the whole cohort with careful annotations of pathogenicity and of penetrance of pathogenic variants across all types of cancer. So we can with fairly high precision look across cancer types and understand the rate of pathogenic germline variants. And you see the highest in nerve sheath tumors with NF1 alterations, and much lower for skin cancers, lung cancers, and so on. But that's really only the beginning. So we can break that down by gene as well. So as I said, nerve sheath tumors typically have germline alterations in NF1. You can see for BRCA1 and BRCA2, I don't know if you can see my cursor, but there are frequent alterations in ovarian cancer, pancreatic cancer, ampullary, prostate, breast cancer. The dark circles represent where there's a statistically significant, or the outlines represent where there's statistically significant association between the gene and the cancer type. And from this, we can observe that 5% of patients, whereas 18% of all patients have a pathogenic or likely pathogenic germline variant of some penetrance, 5% have at least one high penetrance germline variant. But where I think some of the novel analyses can come, where we have this unique opportunity now to look at the intersection between the somatic tumor alterations and the germline variants that are inherited, to ask, well, when we have a germline variant, what's happening in the tumor? Is that germline allele retained? Is the wild type copy lost through biallelic inactivation? Are there co-occurring mutations or mutational signatures that only arise in the context of a germline variant? Really this intersection where I think we have a lot to learn about the role that germline variants play, not just in cancer risk, but also cancer progression and ultimately treatment. So we can layer on top of that figure the rates of biallelic inactivation of a given gene in a given cancer type by looking at the tumor. And from doing this, and so to make a long story very short, we see a wide variation in the rate of biallelic inactivation across cancer types. And as we study this further, recognize that this rate of biallelic inactivation depends in part on the penetrance of the allele, the specific gene that's altered, even the specific variant that's altered, and in interesting ways, the cancer types that result, where just sort of taking an example, BRCA2 across the top, BRCA2, for the tumor types where BRCA2 alterations are associated by prevalence, the ones that were circled in black, those typically also have higher rates of biallelic inactivation than what we observe for lung cancers or renal cell cancers that arise in carriers of these alterations, where the alteration may exist, but it could be more likely a sporadic cancer within those carriers. So there's a lot of interesting biology then to skip over. Overall, we see a few different sort of classes of cancers. This is sort of meant to capture some of the trends that we observe in the data. So in black, we have the germline carriers of high penetrance alleles, and most often those resulting tumors are in cancer types that are associated with those alleles and exhibit biallelic inactivation. Whereas in carriers of moderate or lower uncertain penetrance alleles, almost all of those cancers are in unassociated cancer types and retain heterozygosity of those alleles. And still 27% of the cancers that arise in high penetrance germline carriers are in non-associated cancer types and exhibit heterozygous rather than biallelic status of those variants. And the cancers themselves typically exhibit different clinical and somatic phenotypes, where the cancers where there is biallelic inactivation in associated lineages arise at younger ages with fewer somatic oncogenic drivers, and for whom we detect co-occurring mutational signatures. And this sort of dichotomy can be observed even in a single patient. So we, in this example, observe independent cancers, independent primaries in the same patient that can themselves show differential dependence on the germline allele. So there's a patient with Lynch syndrome because of a germline variant in MSH6 from the DNA mismatch repair genes. And when you look at the colon cancer versus the bladder cancer in this one patient, you find that the colon cancer is heterozygous for the germline MSH6 mutation, has a small number of additional mutations, including HER2 amplification. Through IHC, MSH6 staining is retained, whereas the bladder cancer exhibits an MSI signal, lots of additional mutations, loss of the MSH6 protein, and an MSI phenotype. So, I think even in the same patient, you may exhibit different mechanisms of biological progression of cancers, which may or may not depend on the inherited germline allele. So, we've also begun, as I mentioned earlier, to look at the germline contributions to clonal hematopoiesis. It's another somatic phenotype, and we can ask with our cohort whether the rate and the magnitude and the mutational spectra that we observe in patients with clonal hematopoiesis can be determined in part by the underlying germline alleles. So we can take the nearly 30% of patients that have clonal hematopoiesis mutations and the nearly 18% of patients that have pathogenic germline variants and look to see if there's any statistical enrichment. And there is, nominally significant, but p-value less than 0.01, even after controlling for age and gender and race and smoking and therapy, where there does seem to be an enrichment of clonal hematopoiesis in germline carriers of pathogenic germline alleles. Obviously, this is ignoring sort of broad GWAS signals or polygenic risk scores or other sort of haplotypes that may be associated. So just looking specifically at the same pathogenic germline alleles that we know are associated with tumor progression. We're exploring this more. The signal seems to be potentially coming from certain susceptibility genes and not others, but this is sort of an ongoing investigation that we're pursuing in the lab. But obviously, as many of you appreciate well, there are many limitations of tissue-based sequencing. So first of all, we can't escape the standard histopathological evaluation that pathologists have been performing for centuries, in part because the morphology is still such a critical component for diagnosing and staging cancers. But also it's needed to identify tissue suitable for molecular analysis and interpreting molecular results from tissue-based sequencing. The tumor sequencing panels like MSK-IMPACT, while they detect some gene fusions, they may not detect all. They're not that well suited, given that a breakpoint can occur in intronic or intergenic sequence that might not be included in the panels. By definition, by looking at just DNA, it can't detect gene expression levels. And these are most directly addressed through RNA sequencing applications, which I'm not going to be speaking about today, but which we've been pursuing. But because cancer can be heterogeneous and a single biopsy might not be representative of a patient's disease, or tissue might not even be available for molecular analysis, you can't get a biopsy, or it might be too risky. This is where cell-free DNA from plasma and other fluids has significant potential to overcome some of these limitations of tissue-based sequencing. And we've been doing cell-free DNA profiling for many years from cerebrospinal fluid. So what we found is that in cerebrospinal fluid collected from patients either with primary brain tumors or with solid tumors, lung cancer, melanoma, that have metastasized to the central nervous system, we can detect tumor-derived cell-free DNA in the CSF. And about 50% of the time for brain tumors, about 2/3 of the time for metastatic cancers to the CNS, we can detect tumor-specific alterations. And for that, we actually use MSK-IMPACT the same way we do for tissue, because when we are able to isolate DNA from the CSF, it's typically a fairly high tumor fraction. There's not really much background, which allows the same methods that we've developed on tumor to be equally applicable. So for CSF sequencing by MSK-IMPACT, we received New York State Department of Health approval over 2 years ago and have now sequenced clinically and reported out over 400 CSF cell-free DNAs sequenced. And this work was led by Tejas Bayal, Maria Arcil, and Rima Benaid in our molecular diagnostic service. But with plasma cell-free DNA, what's sort of more common and you're probably more familiar with, this is in some ways even more exciting because it holds many advantages over tissue and is easier to obtain than lumbar punctures that are a common source of cerebrospinal fluid. So it's less invasive than biopsying and LPs. It's typically cheaper to obtain, easier to obtain. Tissue might not always be available. And in theory, rather than relying on a single biopsy, the cell-free DNA may better capture the heterogeneity of the patient's cancer. And the ease of collecting blood facilitates longitudinal monitoring of disease burden and acquired mutations as the tumors evolve or develop resistance to therapies. And compared to tissue-based DNA, there's a much broader range of potential clinical applications. So there's the standard genomic profiling, which is pretty much what we're always doing when we're sequencing tumor, just to identify potentially targetable alterations or genomic features or signatures in cases, and using plasma when tissue fails or we can't obtain a biopsy. But we can also use it for longitudinal monitoring for drug response, tumor evolution, drug resistance, I mentioned. And earlier in a patient's disease course, use it as a means to detect minimal residual disease after surgery or surveillance of cancer recurrence. And even as you're all well aware, before a diagnosis, using cell-free DNA as a means for early detection of cancer. The challenge that all of these applications share is that in plasma, the tumor fraction is typically really low. With some exceptions where patients have a very high disease burden and might have large amounts of tumor-derived DNA in the plasma, we typically need to be able to detect mutations at a much lower level than what we would otherwise require, which then requires its own molecular and bioinformatic modifications to the analyses. And a lot of this work has been pioneered at Stanford, and I'm sure many of you are working in this area and familiar with methods. But as other companies and academic labs have done, we have pursued and incorporated molecular barcoding to suppress these background sequencing errors. So just to sort of restate the problem, if we need to be able to detect mutations down to or below 0.1% allele frequency, it's a big challenge when this background sequencing noise is sort of confounding mutation detection in that same range. So noise can come from DNA damage, can come from enzymatic errors during polymerase PCR amplification, and sequencing errors themselves. making it difficult, this is real data shown here, making it difficult to call mutations in that range with high confidence without introducing all those false positive mutations. So how can we reliably call low frequency mutations within this noise range? We remove the noise. And how do we do that? This is, shows suppression of, through our data, suppression of the background noise by 3 orders of magnitude. We incorporate molecular barcodes. So the work that I'm going to show is was developed and then translated by members of our Technology Innovation Lab in the Center for Molecular Oncology, led by Brian Loomis, who's been working on this project for a number of years. So the barcodes that we use are duplex UMI adapters that allow for tagging of both strands of the cell-free DNA molecule. Probably either done work in this area or heard talks in this area, so I don't need to go into too much technical detail. But the basic premise is that these barcode adapters allow for recognition of replicate sequencing reads from a PCR product that were derived from the same original template DNA, and for distinguishing reads generated from the top strand from those that are generated from the bottom strand. So if we take a single duplex molecule of cell-free DNA, amplify it, and then and collapse those replicates into a single consensus sequence, we can recognize those duplex families where we have replicates from the top strand and replicates from the bottom strand, which allow us to recognize and filter out the background sequencing errors, as well as strand-specific damage that may only be specific to one of the 2 strands, and develop a consensus sequence that eliminates those background errors. For us, the consequence of sequencing to such deep coverage is that in order to keep costs sort of affordable and sustainable, you have to reduce the size of the panel. So we took the content from the 505 genes that we're sequencing with MSK-IMPACT, and looking back at the time over about 25,000 tumors that we had sequenced, selected the most informative regions from MSK-IMPACT for further sequencing. So these were the most frequently mutated exons, as well as sites harboring potentially targetable alterations, carried over additional content that allows us to call microsatellite instabilities, allele-specific copy number, and the most frequently rearranged genes to detect structural variants as well. So altogether, we came up with a panel called MSK-ACCESS that's 13% as large as MSK-IMPACT, but captures 47% of all mutations that we've reported by MSK-IMPACT. So while we had to focus down, this gives us the highest yield of what we can detect. And as of 2019, 2 years ago now, we have been running this as a clinical test. For every clinical test, I may have mentioned early on, we have to first individually validate the assay per New York State Department of Health regulations to get approval to run it as a clinical test. So it's sort of a high bar for each assay, but they undergo a fairly rigorous analytical and clinical validation to get to that point. So after having gotten to that point with MSK-ACCESS, and happy to point you to the validation data that we've released, we've now sequenced over 3,000 plasma cell-free DNAs from over 2,500 patients within our clinical lab and Department of Pathology. And like we do for MSK-IMPACT, we release those results in real time to MSK investigators in the de-identified data set in the CBio portal. They also get reported back to patients and their doctors to inform treatment decisions. And from these 3,000 samples, we've detected at least one alteration, about 73%, with a median of 3 per patient. So smaller than IMPACT, obviously, it's a more focused panel. 41% of samples have at least one targetable alteration. That's partly due to the enrichment of lung cancers within this data set, which tend to more often harbor targetable alterations than some other cancer types. And the variant allele frequencies that we've observed are as high as 99%. These are mutations that are typically amplified and occur in patients with a high burden of disease, and as low as 0.02%. But the median across all mutations we've reported, 1.9%, is actually just below the lower limit of detection of MSK-IMPACT. So these are, the majority of mutations would have been missed by conventional sequencing using the tests that were available at the time. I want to mention, and this has implications for early detection as well, the importance of sequencing the matched normal white blood cells. Because if we just sequence the plasma, we'd be at risk of calling and reporting not just germline variants, which for the liquid biopsy applications are often a little bit easier to detect because the fraction of tumor tends to be low enough, and an heterozygous germline mutation is less often confused for a true somatic mutation. But more importantly, mutations associated with clonal hematopoiesis can really confound the methods most widely used commercially, actually, that sequenced just plasma cell-free DNA alone, with a high risk of reporting back mutations associated not with the cancer, but with normal clonal pseudo-normal clonal hematopoiesis processes, and can be misattributed to cancer and have significant implications for diagnosis and treatment. So here's looking at the data from the first 1,000 or so samples that we had sequenced using MSK-IMPACT, where the variant allele fraction of the mutation is shown on the x-axis, and so variant allele fraction in plasma is shown on the x-axis, and the variant allele fraction in the white blood cell normals is on the y-axis. And the mutations that we actually reported and infer as somatic mutations coming from tumor are the ones in red here. And they also exhibit the property, if you look at the fragment lengths of the DNA molecules that harbor these mutations, to have a shorter fragment length on average than the wild type harboring fragments at those same sites. And this is because of now well-established principles or observations showing that DNA derived from tumor cells is shorter on average than white blood cells due to linker histones and exposure of DNA to damage. Pretty easy to detect germline variants. They're present about 50% levels in both the plasma and the white blood cells. And if you look at the fragment size distribution of the fragments harboring the variant versus wild type, they're superimposed on each other, suggesting that the source of these variants is not tumor cells, but white blood cells. Although I would point out that a number of the somatic mutations from tumor are in the same variant allele fraction range. So we can't just look at the plasma and mask out anything in the 40 to 60% range, because we'd be missing a number of true somatic mutations there as well. But what I wanted to point out most are these clonal hematopoiesis mutations, which are at lower variant allele fractions, typically what you expect the most often in tumor-derived mutations in plasma, but they're also detected in equally high levels in the white blood cells. And if you look at the fragment size distributions, they're more similar to what we expect from germline alleles, suggesting that they are coming from white blood cells as well. And that's a large number of mutations that, in the absence of white blood cell data, could be incorrectly misattributed to cancer when you're just looking at plasma. And this has been, we have now a large series, and I'll just show two, of anecdotal cases where commercial cell-free DNA testing has shown, I think, incorrectly, the presence of mutations, which are really just due to clonal hematopoiesis. So here's one example of a patient with gastric cancer with an MSI-H tumor, so 64 mutations detected by MSK-IMPACT. But when the commercial cell-free assay report came back, none of those 64 mutations were detected, but this one mutation in TP53 was detected about 1.3% variant allele frequency. And if we go back and look at the matched normal white blood cell data from MSK-IMPACT, we found that there was a clonal hematopoiesis mutation in TP53 in about 1.8% of reads. So this was clearly clonal hematopoiesis. It's not associated with a targetable treatment, but just the fact that this report comes back positive could be incorrectly interpreted as evidence of disease recurrence, leading to interventions that might not be appropriate in this case. Here's another patient with a different commercial assay with 58 mutations detected in their colon cancer by MSK-IMPACT. None of those 58 mutations were seen in the commercial report, but there was one ATM missense mutation that was reported. Not only was this established to be clonal hematopoiesis from our MSK-IMPACT data, but there were treatment recommendations that were proposed, suspicious treatment recommendations that were proposed based on the presence of this allele that wasn't even present in cancer. So I think this is a big problem, and it's beginning to be recognized, but I think it's going to continue to confound the liquid biopsy testing for detecting targetable alterations, and a major challenge for using liquid biopsy for early detection of cancer when the signal might actually just be coming from clonal hematopoiesis. Just to show you some of the results that we've been analyzing from the MSK-ACCESS platform, One of the other common phenomenons that we're observing is polyclonal acquired resistance after treatment with certain targeted therapies. This was a study we recently published of a small set of 8 FGFR2 fusion positive cholangiocarcinomas where we had sequenced plasma using MSK-IMPACT, MSK-ACCESS, both before and after treatment with FGFR targeted therapies. And in this cohort, 6 of the 8 patients had acquired mutations in the FGFR2 gene, not present with the fusion before treatment, but emerged on treatment. 5 of those 6 patients had multiple independent mutations acquired in FGFR2, with 2 of the patients having 11 and 13 independent FGFR2 mutations, polyclonal contributions to the single plasma sample. And you can see here just one of these patients where the FGFR2 fusion was detected, then decreased during response to treatment, and then reemerged with the acquisition of, in this case, 11 additional mutations in FGFR2. Here are 2 more examples. One was published in a study where patients were treated with TRAC inhibitors for their NTRK fusion positive tumors. And this patient actually initially developed an MET amplification acquired on treatment with a single agent TRAC inhibitor. And then after the addition of crizotinib, which is a MET inhibitor, the patient acquired 13 different independent mutations in MET. And here's another case of a BRCA2 germline carrier prostate cancer, where after treatment with PARP inhibitor, acquired 7 independent reversion mutations in different subclones that were all detected in cell-free DNA, all restoring the reading frame of BRCA2 from the germline 2-base deletion. Sorry. And so as we move forward, we're doing now work to further increase the sensitivity for detecting circulating tumor DNA, because while we have an assay that's proving to be useful for genomic profiling of metastatic patients for treatment selection, there are those other applications that I mentioned where we'd love to be able to use similar methods for response monitoring and even minimal residual disease detection based on a tumor profile, and using those mutations that are known from the tumor as a basis for serial sampling and monitoring of recurrence of disease. And also for early detection, where even without knowledge from the tumor, can we identify signals that are derived from tumor DNA, either mutations or fragment length or methylation patterns. And here, we're pursuing a number of different approaches in my lab and the CMO Innovation Lab, where, through new versions of MSK-ACCESS with more content, we can increase our chance of detecting alterations. But taking prior tumor profiling data using exome or genome sequencing, designing patient-specific panels similar to Natera's approach, harnessing global copy number data from low-pass genome sequencing, or the size or locations of fragments and where they map to the genome. And we have some work that is still in its early stages, adapting methods for cell-free DNA methylation. It might be useful here. What I want to just spend the last few minutes discussing is what we think is going to be a promising application, as I said, in patients with known high risk of developing cancer, because their lifetime risk is so much greater. And this is specifically for patients with Lynch syndrome. We have some grants primarily with Sophia Stadler to pursue this cohort of patients. Sophia is an oncologist in our GI service as well as clinical genetics, who specializes in patients with Lynch syndrome, where with this registry of patients who are serially monitored, can we use cell-free DNA to detect the early onset of cancer, where we know these patients are at greater risk of developing cancer, and that the tumors that they develop, because of the inherited mismatch repair deficiencies, are likely to exhibit specific patterns. So these patients typically have tumors with lots of mutations, so more to detect, as well as microsatellite instability signatures that we can specifically look for. So in order to do this, we have been working for a couple of years now on improving our ability to detect microsatellite instability from cell-free DNA. And here's Preeti again. So when she was here as a graduate student, she was driving this project with Sophia and Lucy Latham, who was a clinical geneticist, applying our methods for cell-free DNA specifically for patients with Lynch syndrome. And a precursor to this study was a survey of patients with Lynch syndrome, a survey of all patients who had MSK-IMPACT profiling, and looking at the proportion of those who had mismatch repair deficient phenotypes or microsatellite instability signatures detected in their tumor, and the proportion of those that actually harbored underlying germline variants associated with Lynch syndrome. We found that that was highly variable across cancer types. I think it was 16% of all microsatellite instability high tumors, but 0.1% of all microsatellite stable tumors harbored variants associated with Lynch syndrome. So how can we use this information to detect microsatellite instability from cell-free DNA? So Preeti developed a method called ADME, where it's still unpublished, but we're generating a lot of data, and I'm really excited about this method, where you can look specifically at microsatellite sites, like we do for MSK-IMPACT, and look for shifts in the allelic distribution for insertions and deletions at the specific microsatellite sites that differ between plasma and the matched Buffy code. So this would be an example of a single microsatellite locus at a single patient. And if you take the distribution of all microsatellite loci, on the left, it would, and compare, the distance vector between the plasma and the Buffy code, and look at the distribution of those distance vectors in a patient with a microsatellite stable cancer on the left versus a patient with a microsatellite unstable cancer on the right, you can use these greater differences between plasma and Buffy code as evidence of microsatellite instability. So you can look at these distribution of distance metrics for patients with MSI high cancers versus microsatellite stable cancers, and through development of an SVM, set a boundary, and above that, identify patients who have microsatellite unstable cancers. So we're further developing this method, but we've applied this in practice through our clinical MSK-IMPACT sequencing, and used it to identify patients with MSI high tumors that we wouldn't have necessarily expected. This has been a recurring story from MSK-IMPACT sequencing, and now MSK-ACCESS as well. And here's an example of a small cell lung cancer, a patient with small cell lung cancer, where we don't typically observe MSI and wouldn't typically test for this in standard pathology workflows. But the MSK-ACCESS data revealed a number of mutations and also a strong signal for microsatellite instability. This was subsequently confirmed by an MSI-specific PCR assay. As a result of this MSK-ACCESS result, the patient was started on atezolizumab immunotherapy a few days later, and remains on treatment after 9 months of therapy. So this is where we're looking to take this, and with Sophia's through Sophia's collaboration, we're collecting a large cohort of MSI positive cases, both with Lynch syndrome and sporadic cancers, in part to understand if we're going to develop methods for early detection, we want to know what that baseline looks like as well for patients with Lynch syndrome that are unaffected with cancer, in case the presence of these germline alterations could actually lead to detectable signal that we'd be observing, not from a cancer that arises, but maybe from white blood cells. So these are creating a lot of hypotheses that we can test, and I think an exciting area for development. So I wanted to wrap up just sort of summarizing what we've done and where we're going. We've invested heavily as an institution on our clinical genomics program and sequencing tumor tissue. And as we move forward, we want to incorporate not just more genes, but also other genomic content. Our latest panel has viral DNA to look for virus-associated cancers as well, and expand significantly the breadth of what we report and what we can infer from the data itself. So not just the presence or absence of mutations, but signatures, genome doubling, zygosity, clonality, and integer copy number. We acknowledge that the sort of demographic distribution within our cohort is not representative of the population of New York City, let alone the country, and more representative of the patients who come to the Upper East Side to have cancer care at Memorial Sloan Kettering Cancer Center. So we're working on expanding the population to early stage cancers, but also importantly to underrepresented and underserved populations through partnerships with community-based hospitals here. I think this is the year where I feel like we're ready to move beyond panels to whole genome and whole transcriptome sequencing, but with the required analytical validations that take a lot of resources and manpower, as well as the clinical benefits that we've realized from the panel sequencing. We just haven't quite gotten there yet, but panel sequencing has taken us this far. But I'm sure many of you were excited to move to more comprehensive assays. And then the opportunities in liquid biopsy I've discussed, not just for plasma, but urine, cerebrospinal fluid, effusion fluid, other types of fluid, for not just genomic profiling, but for surveillance and response monitoring and minimal residual disease detection, and ultimately for cancer screening and early detection. So I want to, as sort of briefly as I can, and as insufficiently as it may be, acknowledge the contributions of a huge number of people within our molecular diagnostics service, from lab technicians to clinical fellows, clinical bioinformatics team, and leadership of bringing these clinical assays forward to produce these data sets that we've been able to benefit from for research, as well as members in my lab, our Center for Molecular Oncology, and other collaborators at Sloan, Kettering, and funding sources that have enabled this work, that have really been a pleasure to work with, and you know, the work is far from done, as I'm impressed by so much of what I'm hearing from, you know, meeting with many of you, the sort of real cutting edge technology development that's happening in Stanford. Maybe we can look at what you're doing and try our best to implement some of those methods here at Sloan Kettering. So thank you very much. It looks like I'm a few minutes under, so I'm very happy to stay and answer questions if there are any. Thank you. I'll stop sharing my screen too. And I see some, actually I see a Q&A box and I see a chat box. So I will read the question in the Q&A box from Catherine. So many clinicians order the Foundation 1 test, even when their institution has a lab developed test, which does not report the germline results. What are the implications of this? And what does the future hold? A related question is whether plasma cell-free DNA tests will become increasingly used even when tissue is available. Lastly, are you having any issues with payers covering tests? So thank you for all the great questions. I'll take them one at a time. So I guess insourcing versus outsourcing. Yeah, I am aware that many of the academic centers are using commercial tests, and I think that serves an important purpose. I don't want to sort of disparage the commercial testing that's happening. I think a lot of the incentive there is just a smooth workflow where it's easy for a physician in their busy clinic to order a test. And a lot of institutions have built up processes to streamline Foundation ordering or Tempus ordering or other commercial tests that are available. For us, it was a major investment, but I think it's been really valuable for the sort of breadth of clinical data that we can detect and report back. So the ability to include within our reports information about zygosity and other signatures. I don't think we'd have been able to do that or sort of move the needle if we were outsourcing to commercial labs. So that's been of value to us. And also the research that's been possible. So, I think I recognize I'm very lucky to be at a cancer center that invested heavily in this area, but I think the investment has paid off in the ability to expand our clinical genetics research program and initiate the first of its kind clonal hematopoiesis clinic and develop programs around cell-free DNA where we have the tumor raw data that we can use to guide the development of cell-free assays and as a baseline for disease monitoring or other early detection, early intervention type methods and programs. So I think that's been really great for us. I don't regret any of that, and I'm lucky that we've invested in this area, but I do recognize that some of it's just inertia and the sort of challenges of clinicians in changing their clinical patterns and ordering patterns and workflows. So the next question, plasma cell-free DNA, will that be used where tissue is available? I'm definitely seeing that starting to happen. I think lung cancer oncologists would much rather order a liquid biopsy test, whether it's from us or from a company, because they can do that directly. It doesn't require tissue. It doesn't require the involvement of pathologists, which can add delays. But I would push back and say that they're performing a very important role in the diagnostic process. But it also is quicker. The liquid biopsy tests can be, the commercial ones returned within 5 to 7 days. And that could be within a treatment decision window that if you had to wait 2 or 3 weeks for a tissue-based test, you might pass. So the thoracic oncologists here most acutely, but other oncologists as well, are interested, definitely enthusiastic about liquid biopsy tests because of the not just ease of ordering, but speed of results where that can actually matter. So I can see that happening, but for us, or at least in my opinion, tissue is still the gold standard and what we're encouraging whenever possible. And last, are we having issues with payers covering our tests? It's hard for me to answer that. I think that sort of reimbursement data is to some degree obscured from all of us in pathology, even the chair of pathology. It's part of the overall operating budget of the hospital. And I think what our finance department chooses to pursue involving, related to reimbursement is a financial decision that the institution makes. I think they're going to work harder to get full reimbursement for a procedure or surgery than a molecular diagnostic test that's performed. So I don't think we're recovering all of our costs, but when it's viewed within a much larger operating budget for the hospital, I think there's and the benefit for the clinical trials and clinical research program. I think that's been successful and something our institution has been able to absorb. But largely speaking, we're getting much better reimbursement for our tissue testing now with MSK-IMPACT than we did a few years ago. But we haven't really done even much. We haven't really pushed it very hard for MSK-ACCESS. Outside of lung cancer, all of the clinical MSK-ACCESS testing that we're doing is paid for using institutional research and philanthropic funds. I hope I answered that sufficiently. Next question, how deep do you sequence plasma and match Buffy coat? Good question. Sorry, I didn't mention that. So the plasma is sequenced to a raw depth of 20,000x coverage, and that gets collapsed down to about 1500x to 2000x duplex coverage. This is with an input of about 20 nanograms of cell-free DNA. So we use the duplex collapsed coverage for mutation calling because that has the sort of the greatest amount of error suppression, the highest fidelity data. But we still use the uncollapsed data for calling copy number alterations and rearrangements, where the collapsing doesn't really, matter quite as much. For the matched Buffy coat, we're sequencing to about 1 to 2000x raw coverage, so not the same as the plasma. We don't do the same level of collapsing. This is a potential limitation for calling low-level clonal hematopoiesis mutations with high confidence, but we still feel pretty good about our ability to use that Buffy coat data to filter out clonal hematopoiesis mutations that might be low levels. But when we see a mutation in plasma after collapsing at 0.1% or less than 0.1%, and we don't see it in the Buffy coat, we can't prove that it's not a clonal hematopoiesis mutation that's at a very low level that just happens to be present in a few molecules in plasma and not at all in Buffy coat. So that's still a challenge, but I think that's a challenge even for an assay that would sequence the Buffy coat to the same really high depth and collapse, because you're still, when you're dealing with individual molecules, there's always the risk that it happens to be present in one tube and not the next. So that's sorry, maybe more long-winded than the question called for, but thank you for the question. Anything else? No.
Questions for Mike? Go ahead and type it in the Q&A or raise your hand. I can call on you. Oh, Johannes, please go ahead.
So I have, can you hear me?
Yes, we can hear you. Thank you.
I have a quick question in terms of if there are any thoughts on when you do treatment monitoring with the cfDNA, how frequent should you do it compared to imaging treatment monitoring?
I think that, I'm sure you have, strong insights there as well, and may be able to even better answer that than I can. But I think the hope is that cell-free DNA profiling can be done much more frequently than imaging has traditionally been done. I mean, there could be innovations in imaging that might, that might change that. But, I think we're actually, and I'm not a clinician, but I would all appreciate that we're living through a transformation in care delivery, and a lot more things will be done remotely in the future than that would have in the past required an in-person visit. So if we can be even collecting blood remotely rather than bringing patients in every however many weeks or months for expensive imaging, that might further underscore the benefits of liquid biopsy testing. So I don't know the answer sort of from a clinical or scientific standpoint, but I think even just from a practical or logistical standpoint, the liquid biopsies might fit better in future care paradigms than imaging will.
Okay, thank you.
Excellent. Any other last questions from anyone? Okay, well, thanks so much for a really excellent talk and discussion, very insightful, a lot of really great future directions, and it's a pleasure to having you.
Yeah, thank you. And again, I regret it can't be in person. It's a little awkward over Zoom, but hopefully it went okay. But thanks to everybody who attended and for the invitation.
All right, thanks everyone. Have a great day.